Hello friends, welcome to VMH Learning. In this video, we will discuss about agglutination inhibition test. Commonly, it's also called as pregnancy test. So, you would be able to know how pregnancy kits work. Before we get into the concept of agglutination inhibition, I would like to tell you about the basics of agglutination and precipitation. We know the RBCs are large and particulate antigens. So, when these large antigens are mixed with their specific antibodies, what you would observe? These antibodies will bind to the RBCs. The binding we are seeing here is in a specific pattern called lattice. And this reaction is actually visualized in the form of clumping. Now, you have to notice one thing here, that this clumping is occurring within few seconds after the antibodies are mixed with RBCs. You must have encountered these kind of reactions like in blood group detection tests. These reactions are called as agglutination reactions. Now, if the antibodies are gonna bind to small and soluble antigens in place of large and particulate antigens, what would happen? You will still observe the binding between antigen and antibody. But the reaction doesn't proceed instantly. It requires at least 24 to 48 hours for reaction to become visible in the form of precipitates. These kind of reactions are called as precipitation reactions. So remember this, small and soluble antigens can't be detected quickly like their counterparts. Now this is really important. Using agglutination inhibition method, we can detect very small and soluble antigens very rapidly, even when they don't show agglutination. The principle of agglutination inhibition test was used in old pregnancy test kits. So let's discuss the principle with that example. Pregnant women secrete a hormone called HCG in their urine. Now, this HCG is a very small and soluble molecule. In some books, you will find it written as hapten, since it's antigenic and not immunogenic in nature. So, being small and soluble molecule, the hapten will not show agglutination with its specific antibody. But the agglutination still can be achieved. How? So, when the hapten is conjugated with a carrier molecule, it becomes hapten carrier complex. Now, this hapten carrier complex, if you see, has become quite larger, and thus, when it binds to its specific antibody, a successful agglutination reaction is carried out. So, you have to remember two things. HCG alone cannot show agglutination, but its complex with carrier can show agglutination reaction. Now, we need to know about the reagents used in pregnancy test kits. So there were mainly two reagents. First is the anti-HCG antibodies. Now, these antibodies can bind to both free HCG molecules as well as HCG molecules complexed with carrier molecules. The second reagent is HCG carrier complex itself. Now, in order to understand the principle, we have two scenarios. First is, suppose woman is pregnant. That means the urine sample of the woman should be having HCG. To carry out the pregnancy test, first thing we do is to incubate the urine sample with anti-HCG antibodies. And we know this incubation will cause antibodies to bind with HCG molecules. In the next step, the second reagent, that is HCG carrier complex, is added into the mixture. Now what would happen? We know the antibody can bind both free HCG as well as HCG carrier complex. Since HCG molecules were already present in the urine, so all the antibodies added got engaged with them. So therefore, no antibody was free to bind with HCG carrier complex. Now what's going on here? The Sag molecules from the urine actually inhibited the agglutination reaction between anti-Sag antibodies and Sag carrier complex. And because of this inhibited agglutination, the test is called Agglutination Inhibition test. So, in case of pregnant women, we will not able to see agglutination. Now let's talk about the second case when woman is not pregnant. That means no Sag molecules in the urine. Similar to the previous case, we will treat the sample with anti-Sag antibodies. As we know, the sample lacks Sag molecules, the antibodies will remain free. In the next step, we will add Sag carrier complex. We all know what will happen. The free antibodies will bind to the complex, and there would be visible agglutination. So what's the conclusion? When there is agglutination, the test is negative. And when agglutination is absent, the test is positive. This was all. Thanks for watching.